next talk is by Tanya Feng from UCLA.
Good morning, everyone. First, I would like to thank the conference organizers for allowing me to be here to share some of my research with you today. So since I started my PhD, I've been interested in understanding the effects of natural selection on linked neutral divergence. This work is currently available on the bioRxiv and is in Praxis PLOS Genetics. To me, the motivation for this work is we want to understand how different evolutionary forces can shape the genetic variations that we observe between members of the same species or different species. I've been focused on the effect of natural selection, particularly on neutrocytes. So by definition, neutrocytes are not under the effect of natural selection. So how could that be? Well, broadly speaking, there's two ways that this could happen. The first is genetic hitchhiking. So imagine that these are some neutrocytes in a population. When a mutation arises that is beneficial and is also linked to one of the neutrocytes, this neutrocyte will quickly reach fixation in the population when natural selection acts on this beneficial mutation. Similarly, you could have background selection where deleterious mutations are linked to these neutrocytes. And these neutrocytes will be removed out of the population when natural selection acts on these deleterious mutations to select them out. So the effects of these processes can be seen unambiguously in diversity data, which is DNA differences between members of the same species. The signature pattern that suggests that natural selection can affect linked neutral diversity is a positive correlation between neutral diversity and recombination. So here at regions of low recombination where loci are tightly linked, these sites are affected more by the effects from hitchhiking and background selection, and therefore you see this reduction in diversity, whereas at regions of high recombination, these loci are unlinked and therefore not affected as much by genetic hitchhiking or background selection. So this pattern was first observed in Drosophila in the early 90s. Other researchers have since observed the same pattern in a variety of species, such as yeast, humans, other model and non-model species. Well, when examining whether natural selection can affect linked neutral diversity, which is DNA differences between members of different species, it becomes less clear. There's a long-standing theory in population genetics that suggests that natural selection should not affect linked neutral divergence. And the way that this theory works is that it only considers the substitutions that occurs after two lineages split. However, there's also an extra component that is also important, which is the substitutions that are found in the ancestral population, which is the population that occurs before the split. So these substitutions, colored in green here, could be affected by genetic hitchhiking or background selection, similarly to the genetic diversity currently today. So this theory assumes that the effect from the ancestral polymorphism is not significant because the ratio between the ancestral polymorphism to the total divergence would be very small. And so we question whether these theoretical results still hold when you consider ancestral polymorphism. Well, we're not the first one to ask this question. When other researchers examined this question using empirical data, what they found was rather conflicting. So some studies found no evidence supporting that natural selection can affect linked neutral divergence. Some other studies found very strong evidence supporting this hypothesis. And so due to these conflicting conclusions, what we wanted to do is to test whether natural selection can affect linked neutral divergence, and we want to do this more systematically and more rigorously using a variety of techniques that I want to tell you about today. A good question is, why do we care? Well, if natural selection can affect linked neutral divergence, and this would mean that there's a greater effect of natural selection across the genome, and this is at odds with the neutral theory, which has been the prevailing view in population genetics for the past several decades. Additionally, mutation rates are often estimated from neutral divergence, So you can imagine that if natural selection affects neutral divergence and we do not account for this effect, a lot of the mutation rates estimate could be inaccurate. So to test whether natural selection can affect linked neutral divergence, we first look at the relationship between neutral divergence and recombination. And we first do this in pairs of closely related species. So we chose human chimp and human orangutan data set. And I want to emphasize here that we use neutral divergence, and in order to obtain these putatively neutral divergence, we apply a variety set of filtering criteria, and I'm happy to discuss that later, how we do that. But in both cases, we saw a strong positive correlation between neutral divergence and recombination, and this signature pattern is consistent with the hypothesis that natural selection can affect linked neutral divergence. A second test that we did is to examine the relationship between functional content and neutral divergence. So here we define functional content as a proportion of sites that are coding or conserved in a region. So if natural selection can affect linked neutral divergence, you will expect to see less divergence at regions with more functional content where more sites are under direct effect of selection. And this is what we saw. We saw that in both cases, we saw a strong negative correlation between neutral divergence and functional content, and this is another compelling evidence supporting the hypothesis that natural selection can affect linked neutral divergence. An obvious question is whether these patterns also hold for distantly related species. So we chose to use human mouse and human rat data set, and again, in both cases, we saw a strong negative correlation between neutral divergence and functional content, consistent with the hypothesis that natural selection can affect linked neutral divergence. Then instead of looking at recombination, because recombination rate is not conserved between human and mouse, we chose to use a different statistic. And we used the estimate of background selection, and these values are called the b-values, and these b-values were estimated from within primates. So there's no a priori reason why these human-mouse divergence should be correlating with these b-values. But what we found was actually there's a strong positive correlation between human-mouse divergence and human-rat divergence to these b-values that suggests that at regions that are experiencing a lot of background selection within primates, these regions also overlap with regions that are experiencing background selection in human-mouse ancestor and human-rat ancestor. So overall, this positive correlation is consistent with the hypothesis that natural selection can affect linked neutral divergence. So far, empirical patterns have been quite compelling at suggesting that natural selection can affect linked neutral divergence, but the conventional wisdom in the field is that natural selection should not affect linked neutral divergence. So we wanted to test whether a neutral model could recapitulate the empirical patterns that we observe. To do that, we use coalescent simulations under a neutral model, and the red line represents the empirical correlation that I showed before, and we saw that none of our simulations could generate a data set that can generate a correlation that is comparable to the empirical data set. But what we saw was that when we incorporate background selection into our simulation, now we saw that these simulations can generate a correlation that is comparable to the data set. We also explored the possibility of mutation rate variation, but we saw that mutation rate variation by itself cannot generate these correlations. So far, both empirical patterns and coalescent simulations are pretty compelling at telling us that neutral divergence could be affected by natural selection, but we want to gain more theoretical insight into this effect. And to do that, a postdoc in our group, Christian Huber, used a simple two-locus model, where the first locus is neutrally evolving. The second locus is experiencing some effect from background selection, where background selection causes the coalescent time to be shorter compared to the locus that is neutrally evolving. And what he saw was that for a particular mutation rate, a particular ancestral population size, and a particular b-value, background selection can explain a big or a significant proportion of the variance in the total divergence across the genome, even when the ratio between the ancestral polymorphism to the total divergence is quite insignificant. So what we conclude from this set of analyses is that even when the contribution from the ancestral polymorphism is small, background selection could still explain a significant proportion of the total divergence across the genome. Well, an alternative hypothesis is that we could be observing the direct effect of selection. So in other words, the neutral sites that we consider are not actually neutral, and they may be under some weak effect of natural selection that were invisible to the filtering criteria that we used. And so to address this problem, we increased our filtering criteria by using GURP score. So these, if you're not familiar, these GURP score are a metric of conservation, and the idea is that a site with a higher GURP score is more likely to be under some direct effect of selection. And so what we did was we recomputed all of our correlation analyses with proportion of the genome removed based on GURP score. And so, for example, what I'm plotting here is the correlation between divergence and recombination in human chimp and human orangutan as a function of the percentage of the genome removed based on GURP score on the x-axis. So what we saw was in most of these correlations, even when we removed 25% of the genome based on GURP score, the correlations remain significant. Only in one case, which is the correlation between divergence and functional content in human mouse and human rat, we saw that direct effect of selection could explain away our correlation only when 15% of the genome is under selection, or under direct effect of selection, which is a higher estimate than what is currently believed, which is about 5 to 10%. So what we conclude from this set of analysis is that in order for direct effect of selection to explain away some of our correlation analyses, it would require more of the genome to be under direct effect of selection than currently believed. So to summarize, I show you some signatures that is in the data that is consistent with the hypothesis that natural selection can affect linked neutral divergence. We use coalescent models to show that models incorporating background selection can recapitulate the empirical correlations that we observe, whereas a neutral model cannot. We analyzed the theory and showed that even when the contribution of the ancestral polymorphism to a total divergence is small, background selection can still explain a significant proportion of the variance in the divergence across the genome. We also explored the direct effect of selection, but in order for this hypothesis to explain our correlation analysis, it would require more of the genome to be under selection, under direct effect of selection than currently believed. And with that, I would like to thank my advisor, Kirk Lohmüller, the postdoc who helped me with the theoretical analysis, Christian Huber, and our funding agencies. Thank you for your attention.
Have time for a few questions? I have a question.
Yeah.
When you estimate the divergence, are you using a single genome per species? Or are you looking at some combined?
Yeah, so I use a reference genome for a single species. Yeah.
Hi, sorry. I liked your talk very much. I'm just wondering what kinds of sites you scored as like neutral sites.
Oh yeah, so in order for a site to be considered neutral, we filter out coding regions, functional regions, estimated from a variety of FASTCons from different phylogenies scope. We also remove all the centromere, telomeres, and the regions surrounding that. Yeah. Oh, you have a question?
Can you explain why it doesn't matter what fraction of the differences occurring as ancestral polymorphism?
Can you repeat the question?
Oh, so your question is why the ratio between the...
ratio doesn't matter.
Yeah, so even though the ratio doesn't matter, it's the variance in the divergence that matter. The variance in the divergence caused by background selection in the ancestral population that matters, not the not the actual ratio. Thank you.
Thank you very much.